Welcome, Master Gardeners. This is Sarah Browning. I'm the Horticulture Extension Educator in Lancaster County. We're going to take the next few minutes and talk a little bit more about plant physiology, morphology, and classification. Specifically, some things about each of these topic areas that you might be able to use as you're managing plants or trying to diagnose plant problems. Let's start off first by looking a little bit more into plant physiology. And you had an extension circular entitled Plant Growth Processes, Transpiration, Photosynthesis, and Respiration that you can refer to for more information on this topic. Let's go a little bit more in depth into photosynthesis and respiration, specifically how these two important chemical processes happen and how they affect plant growth in landscapes. So let's start off with photosynthesis, and as you can see, photosynthesis, plants require carbon dioxide and water for photosynthesis to take place. And photosynthesis can only take place when there is light, so it only happens during the daytime in plant tissues that are green, which has chlorophyll. And through photosynthesis, plants are able to create their own sugar or carbohydrates and oxygen, which they release to the environment. So photosynthesis can be limited by certain factors in the environment, such as light. As I said, photosynthesis can only take place during the day. It doesn't happen at night. It only happens in the tissues that have chlorophyll. So we don't see much photosynthesis taking place in the woodier branches and trunk because there isn't much chlorophyll there. Carbon dioxide is also required for photosynthesis to take place. So if the plant ever runs out of carbon dioxide in the leaves, then photosynthesis will stop, even if all the other environmental conditions are right for photosynthesis to take place. Okay? And sometimes temperature can play into the amount of carbon dioxide that there is in a leaf, and we'll go into this more a little bit later on. Now, respiration is just the opposite process, where plants take the sugars that they have created, and with oxygen, they break those down into components that they can use to grow. So they break their sugars down, and they can create energy that they can use to put on new growth, to create defensive chemicals like tannins, and they can and various other processes within the plant to fuel those processes within the plant. And byproducts of respiration are carbon dioxide and water. So the carbon dioxide is reused within the plant for photosynthesis, and then the water is also used by plants. Now, it's important to note here that respiration is not limited by light. It does not require light to take place. So plants can be respiring or using up stored food energy, whether it's day or night. So as we mentioned, plants have to take carbon dioxide in through openings in the leaf, and they also release oxygen through those same openings. So what we're looking at in this picture, if you look at the bottom side of that cutaway of a leaf, you'll see two little cells which are called guard cells right here and here, and together that structure is called a stomata. You can see another one here, two guard cells and an opening. And plants can open and close the guard cells to either allow moisture to leave the leaf or to prevent moisture from leaving the leaf. And they also use these openings to allow gas exchange. So let's take a little bit more close-up look at those guard cells and the stomata. In this picture, you can see, again, two guard cells. And when those guard cells are fully expanded with moisture, they pull apart and they create an opening which allows moisture and gases to move in and out of the leaf. When those guard cells are dehydrated, they become soft and they fall together and they close up that opening. Now, in the picture above, you'll see an electron microscope image of actual guard cells on a leaf. The stomata and the guard cells are primarily on the underside of plant leaves. And as I mentioned earlier, they allow for water to be released through the leaf surface in a process called transpiration. Water is released and evaporates through the stomates. Now, if transpiration is controlled by several different environmental factors, if it's very humid outside, transpiration will be slowed because there is so much moisture in the surrounding air. The evaporation of moisture from the stomata is slowed. If the soil water is very dry and plants can't pull up moisture very well because the ground is dry, that will also slow transpiration. But if the soil has plenty of moisture, then transpiration will move right along at a normal pace. Again, temperature plays a factor here, which we'll talk about on the next slide, as far as the rate of transpiration in plants as well. So the process of transpiration has several functions in the plant. It serves as a pulling mechanism. As water is lost through the stomata, the adhesion of water molecules to each other, it pulls new water molecules up from the roots through the stem to the leaves. So it helps to move water through the plant. Transpiration also helps to maintain plant tissue turgor pressure. So it helps to keep plants at the right level of water balance. And if there's too much water, then the plant is able to release water through the stomates. If the plant doesn't have enough water, then it will close the stomates that will help to conserve water within the plant. Now, as water is evaporating through these openings, it also serves the important function of cooling leaf surfaces. And plants use this when temperatures are very hot to try to keep their leaves cooler. And so thus it helps to maintain plant temperature. So let's take a look at how plants respond to very high temperatures, which we typically have here in Nebraska, anywhere from July through September. The process of photosynthesis typically shuts down at about 90 degrees Fahrenheit. And so it's important to keep that in mind. If we have very, very hot days above 90 degrees, then oftentimes they are not photosynthesizing because that process has stopped. But if we have nighttime temperatures that are above 70, this is a time when there's increased transpiration and respiration within plants, which is using up energy. So during those hot days of July, August, September, when we typically have very high temperatures and warm nighttime temperatures, we can have plants which are actually functioning at a deficit. They're not photosynthesizing enough to replace the stored carbohydrates or energy that they may have used up during the nighttime. And so it's important to understand this to make sure that primarily we're providing enough water to those plants during those hot temperatures. If plants have enough water to pull or to draw on from the soil, then they may be able to keep those leaves a little bit cooler through the process of transpiration when temperatures are very high. So here in the final bullets you'll see I've listed some of our recent years and how many days above 90 degrees in temperature that we had during those years. So you'll see back in 2020 we had 41 days during the summer when temperatures were above 90. In 2019 we had 22. So it's very common for us to have a lot of very hot temperatures during those mid to late summer months. Another plant response to temperature that's important to understand is how plants can be damaged by large temperature swings that we may have in the fall, like in November, or during the winter months. So here you can see some temperatures the last couple of years that I've recorded for you. So in 2020, January 5th through the 10th, we were having highs during the day in the 40s. January 16th to the 22nd, our nighttime temperatures dropped into the single digits. So it's -1, -4. That's a 40-degree temperature difference. Then in February of that year, we were back up in the 60s. February 13th to the 14th, our minimum temperatures again were -4, -5. Then in the fall of 2020, we had pretty moderate temperatures in the early part of November, with daytime temperatures in the upper 60s to 80s and nighttime lows in the 20s to the 50s. But then in mid-November, the 13th to the 14th, we suddenly had minimum nighttime temperatures in the teens of 13 and 14. So plants were not fully hardened off yet at this point, and were not ready to tolerate those cold temperatures. And this is a situation where we frequently then will have plant damage that will start to manifest itself the following spring. Similarly in 2021, in January 12th to the 15th, we had highs of 41 to 51. Then in February, the temperatures at night got very cold. So the 9th and the 10th, we had minimum temperature of minus 16. February 15 to 17, we had minimum temperatures of minus 15, minus 27, minus 28, so extremely cold. And then in March, the second week of March, we were back up with highs in the 69 to 75. So what happens with temperature swings like this, particularly when they occur in the fall, like in November, is that those cold temperatures will actually kill vascular tissue that is not fully hardened off. And what you're seeing in this picture is a condition we call bark blast, where the cambium has been killed by early cold temperatures. And after this injury happens, usually occurs in about the second year, we'll start to see bark falling away or sloughing off where that cambium has died. And of course, this is a huge injury to this plant. I mean, look at the extent of the bark that is dead, that is no longer functioning to move water or nutrients throughout the tree. So how do we prevent this kind of damage? Well, primarily, since we can't control the weather, we have to just rely on planting trees that are very tough and tolerant, and avoid those species which are susceptible to this type of injury. We see this type of injury very commonly on some of the ornamental pears, the ornamental cherries. We see it in fruit trees. We see it in thin-barked maples. So plant selection is the best way that we have of avoiding this type of damage. Okay, so that was a quick look at some of the physiological processes in trees and how sometimes our environment can cause problems, related to these physiological issues. Now let's take a look at roots. And again, there was an EC associated with this, identifying landscape plants. So there's more information there with a lot of different terms. If you are unsure of a term, you can refer to that EC for a definition. But we know that roots are extremely important for plants. They have so many functions. They anchor plants in the ground. They absorb oxygen, water, and minerals. They're a storehouse for energy or carbohydrates. They can be woody or non-woody. Some roots live for many years, whereas some roots are what we call annual roots. They only live for one season, then they die off, and then new roots grow the next year. What you're looking at in this picture are all of those fine little hairs on the side of the root are root hairs, and they are the primary absorbers of water and nutrients from the soil. Now here at the very tip, we have what's called the root cap, which is an area that's a little more strengthened or suberized, which helps to protect the root and where the cell division is taking place at the root tip, and as that root pushes its way through the soil. So let's look at a few more issues related to tree roots. So one important concept we need to talk a little bit about is where are tree roots located? And this is really important when we're thinking about watering a tree or protecting the roots of a tree if there's going to be some soil disturbance around it, whether that's home construction, renovation, trenching for an irrigation system, utility work near your trees. But where are the roots located? And we know now that roots are wide and flat, like a pancake, and not as deep as we used to think, like when I was back in middle school and going through a biology class. So in this image, what you're seeing is an estimation of the location of tree roots by a tree researcher from the Georgia Warnell School of Forestry, Dr. Kim Coder. So what he estimates is that about 40% of the tree's roots are located under the drip line of the tree. And the drip line is defined as just the widest branches of the canopy, and then if you go straight down from that to the soil all the way around the tree, that's the drip line. So let's just say in this particular instance that the distance from the trunk to the edge of the drip line on this tree is 33 feet. So 33 feet from the trunk all the way around the tree in a large circle, that would be the first 40% of the tree's root system. Now the second 40% is another 33 feet out from the drip line. So now we're 66 feet from the trunk all the way around in a circle. That encompasses about 80% of the tree's root system. The remaining 20% is even farther beyond that. So it's important to understand just how wide and extensive tree root systems can be. So we know that for roots to grow well, they require oxygen from the soil, they require water and minerals for the roots and the tree to grow. Growth of roots can be shallow. As I said, about the top 90% of the tree's water absorbing roots are going to be in the top 24 inches of the soil. So that's much shallower than what we used to teach. And the growth of those roots in the surface of the soil can be limited by oxygen content and by the physical conditions of the soil itself. So if the soil itself is very heavy and compacted with very small pore spaces and little oxygen content, this is going to have an impact on tree growth and tree vigor. So it's important to remember that tree roots grow in the air spaces between soil particles. And if those air spaces are very, very small, that is going to make it more difficult for our tree to grow and to be very healthy. So anything we can do to improve soil quality or soil structure, whether it's aerating, all of those things are going to be helpful for the tree's growth. So let's look at a concept called the critical root zone, or the CRZ. And this is a concept that comes out of what are the roots that you really need to protect to keep a tree alive if you were going to be doing some kind of construction around a tree. So typically one rule of thumb that is given in the industry is that for every for every inch of trunk diameter measured at 4.5 feet up from the soil line, that translates into 1 foot of radius on the soil that needs to be protected for the critical root zone. Okay, so we look here at this tree. This tree has a 21-inch diameter, or the width across the trunk. So we would want to use a 21-foot radius out from the trunk to establish a critical root zone. Okay. Now, if you had a really, really high value tree or a specimen tree that you wanted to definitely protect, then you could increase that factor of a 1 foot radius up to 1.5 foot radius for every inch of trunk diameter at breast height. So what is the impact to the tree of not having enough soil to grow? So in this picture, you see 3 trees, and they are in a landscape bed which is getting progressively smaller as the bed comes to the tip. And you can see that that's had a large impact on the growth of these trees because the canopy of the tree are getting progressively smaller as we get into a smaller amount of soil availability. And you can see those 2 trees at the tip are also really struggling because they're probably not able to pull up enough water. And so either we've had some leaf scorch and some leaf dieback, or we just have these 2 trees that are dying outright. So it is important for a tree to have an adequate soil volume to grow in for that tree to be healthy. And having a tree grow in small restricted soil areas will have a definite dwarfing effect on the tree. Now on the tree at the right, you see a tree that's growing in an area where it's got plenty of soil volume in which the roots can develop. And so this tree consequently is much healthier. Another important concept related to the growth of plants and their roots are mycorrhizal fungi. So this type of fungi is a beneficial fungi that forms a symbiotic relationship with plant roots. So in this picture, these large structures here are plant roots, and these very fine hair-like structures are the roots of the mycorrhizal fungi. So the way the fungi and the tree roots work together is that the mycorrhizal fungi can grow very quickly and at a lower cost in terms of energy and inputs required than a tree root would need to grow. So the mycorrhizal fungi can grow very quickly through the soil, and they can pull up water and nutrients, and they pass some of those nutrients and water back to the tree roots. In turn, the tree roots will give the fungi some of the carbohydrates and sugars that the plant has created through photosynthesis. So it's a very beneficial win-win situation for both the tree or the plant and the mycorrhizal fungi. Now, some estimates are that mycorrhizal fungi can expand the reach of a tree's root system by 500% or more. We're seeing more and more products coming onto the market now which contain ground up dried mycorrhizal fungi, and you can put them in your soil and to help a tree and to help encourage this association between the mycorrhizal fungi and the tree roots. Now, if you're living in an area where you have an older soil, like 25 or more years old, and the soil has not been disturbed, there's probably a good level of native mycorrhizal fungi in that soil. But if you're living in an area with disturbed soil or in a new construction home, then probably the amount of mycorrhizal fungi in that soil is lower. And so the use of a mycorrhizal fungi inoculant to the roots could be beneficial and could help your plants grow better. Okay, so let's shift our focus now and talk for just a few minutes here about flowers. And again, in the Extension Circular on Identifying Landscape Plants, you can find some good diagrams that show you some of the shapes of different flower structures and some of the common terms that we use related to flower structures. So let's look at a few of those terms here. We use the terms complete and incomplete related to flowers. So a complete flower is one that contains a stamen, which is the male pollen structure, a pistil, which is the tip that receives the pollen and is part of the female structure, the petals, which you'll know, and then sepals. And sepals are a covering that covers the bud when it's closed. And I'll show you a picture of that here in a minute. Now, an incomplete flower is one that is missing one of these parts. So it could be a flower that has a stamen, a pistil, and petals, but it has no sepals. Or it could be one that has just the female parts and it is missing the male parts. So all of those types of flowers would be considered incomplete. Now, another couple of terms that we use are the words perfect and imperfect. So a perfect flower is one that has both the male and female parts together in a single flower. An imperfect flower is when you have just a male flower and separate female flowers. And that's what you're looking at here in this picture. These are the flowers of a cucumber, and with this flower here, you can see right below the flower there's a tiny little cucumber attached right there. And the same with this flower that hasn't opened yet. We have a tiny, tiny little cucumber right here. That is telling us that those are two female flowers. So they only have the pistil or the female portion. This flower in the middle has just a slim stem or a petiole that attaches it to the underlying vine. This is a male flower. So all of the plants in our cucurbit family, which would be the cucumbers, the melons, the pumpkins, squashes, all of those plants have separate male and female flowers. A few more terms that we run across in flowers are monoecious. Monoecious, the way that I remember that is mono, or one. You have separate male and female flowers on one plant, which is what we have in the case of this cucurbit, these cucumber flowers. Dioecious, di meaning two, where you have separate male and female flowers on separate plants. So, you think about cottonwood, where we have some cottonwoods that will give off all of that cotton in the early summer. Those are female plants. And then we have male plants which have no cotton, and those are typically the ones that we use more for landscapes. The same for ginkgo and ash. They have separate male and female trees. Then polygamodioecious, and all that means is that you may have a plant that has separate male and female plants, but then they may also have a few flowers which are perfect, which have both functioning male and female flowers together. Now again, if you can't remember all of these terms, you can refer to that Identifying Landscape Plants Extension Circular, and you'll find them listed there again. So, looking at this flower, the question is this a female, a male, a female flower, a male flower, or a complete flower? Well, if you look very closely at the second picture, we have male pollen structures here. We have a very thin pistil, which is the female portion, right here. So we have pistils, and we have stamens. We have petals, obviously the showy yellow petals. And then on the underside, we have a sepal. This is the covering that encloses the flower before it opens. So we have a pistil, we have stamens, we have petals, we have sepals, so that would make this a complete flower. Okay, let's change topics again and look at a few aspects of stems that are really important that you understand. So stems have lots of functions. They protect the vascular system within the tree. They're important for carbohydrate storage or food storage. They have the meristematic areas where new growth occurs. They're important for physical support, for gas exchange, plant reproduction. So stems do a lot of different things for plants. Now, so let's look at some of the meristematic tissues and the terms associated with those as far as plants go. So with a meristem is just a site of new cell division, and behind the cell division we have cell elongation. So in plants we have an apical meristem, and an apical meristem is a bud that is right at the tip of the stem. So here in this little side shoot we have an apical bud or an apical meristem right at the tip. Now a lateral meristem can be defined as the buds on the sides. So we also have lateral buds which have lateral meristems associated with them. Okay, here's a few more terms that we need to understand related to stems. The first are herbaceous versus woody. So a herbaceous stem is basically a stem that only lives for 1 year. And we find this type of stem in a lot of the common garden perennials that we have in our landscapes. Perennial salvia, tall garden phlox, garden sedum, peonies. All of these plants have herbaceous stems. Now this is compared to a woody stem, which obviously lives for multiple years at a time. Another important term is the term lenticel. lenticels are another structure in woody stems that allow for gas exchange. And here on this paper birch stem, we see all of these little white structures. These are lenticels. In some plants, lenticels are very easy to see, as in this example. In other types of bark, they're much more difficult, if not impossible, to see. But they should not be confused for some type of a disease or an insect. In this case, this is actually a natural structure. So two more terms that we need to be familiar with, especially when we're talking about pruning, or if we're going to be getting into plant propagation, are the terms node and internode. So in this picture, we have a plant stem which has leaves, and in the junction of where that leaf stem attaches to the underlying stem, or I should call this a petiole, which is actually the correct term. The leaf petiole is attached to the underlying stem. We have a new little shoot that is developing, and this plant happens to have an opposite leaf arrangement, so we have two leaves opposite each other and two shoots opposite each other. So this would be a node. And a node is an area on a stem where the stem has the ability to generate new growth. We compare that to an internode, which is this section of the stem right here between these two nodes. It does not have the ability to generate any new growth. So if we were to make a cut across this stem, what would happen is everything from the cut down to the next node would die, because that stem section does not have any way to keep itself alive, basically. So if we're making a pruning cut, we're typically going to make the pruning cut right above a node, and also with plant propagation, where the place on the stem where new roots will be generated is from the nodes. So we want to make sure that we have some nodes in the soil when we're doing stem cuttings for propagation purposes. Let's look a little bit at how some of these growth structures are arranged within a stem, or in this case a woody tree. So we have an area of what's called the vascular cambium, which is this green area that wraps all the way around the tree, just right underneath the bark. The vascular cambium is also another meristem, in that this is an area where the tree has the ability to, cells can divide and elongate, and new cells are created. Now, the vascular cambium every year will put on new layers of xylem and phloem. The xylem cells are produced toward the inner part of the trunk, and eventually they will become the sapwood, which is still active cells of xylem, and the heartwood, which is an inactive section of xylem, which is no longer functioning physiologically for the tree. The vascular cambium also puts on a set of cells to the outer area, which is the phloem. And the phloem cells are typically only active for 1 year, and then they are replaced by a new layer of phloem every year. So we can have several years of actively functioning xylem, but we typically only have 1 year of active phloem. And the other thing I want you to notice from this picture is how close to the surface of the bark this vascular cambium is. It's not very deep in the tree, and it's very easily damaged when we run into a tree or stem with a weed whip or a mower or anything else that damages the bark. So here again we can see a tree section. The vascular cambium is right in the area of this dark ring that is right beneath the bark. All of the inner rings that we count to give a tree its age, all of these are rings of xylem. Okay, and here again we have an internal heartwood area of xylem, which is no longer functioning. Typically, a tree may have anywhere from 3 to 7 years approximately of xylem, which are actually functioning, in that they are moving water and nutrients and minerals through the tree. Okay, so the xylem, when it's functioning, is actually dead. At this point, it's no longer alive, but the cells of the xylem are stacked on top of each other and almost form a long straw, up through which water and nutrients can be moved. Water that's pulled up by the roots and is moved up through the vascular tissue in the roots, up into the stem, and then out into the secondary branches. There is in some trees, we look at the differentiation between the size of the xylem cells that are put down by the tree throughout the year. And so we can categorize some trees as ring porous versus diffuse porous. We don't really need to go into a lot of detail with that in this presentation, but that is another classification of trees. So the cells of the xylem begin developing in the fall, and then they continue their development in the spring as the tree resumes growth. Now the phloem cells, in contrast, are still alive when they are functioning. They're not dead like the xylem cells. The phloem cells are the cells that move the foods, the carbohydrates and the sugars that the tree creates through photosynthesis. Those are moved in the phloem throughout the tree, to the branches and the twigs for storage, to the buds, and so on. Okay. Now, one other aspect I wanted to point out to you about how trees put on growth, and that is related to development of branches in trees and how a tree can develop strength. So what I want to show you in this picture, the first image, when the tree starts to grow in the spring, the first thing that starts to grow are the buds, then the little twigs, then the bigger twigs, then the larger area of the branches. So those are growing early, and they do cell division, and they put on new growth. So all of this black or darker area that you're seeing is indicating an area of new growth cells on the branch. So you can see that the cells come down the branch, and then they wrap down into the trunk a little bit. And here you can see in the second picture, this is just a front-on version of what we're seeing from the side in image 1, where those new cells are developing on the branch, and then they come and they project down the trunk a little bit. Now, a little bit later in the spring, the cells in the trunk start to grow, and those cells will come, and the new cells in this image are indicated by the lighter tissue. Those cells will come and they will wrap around the cells that were put, or they will grow over the cells that were put on by the branch. So here you can see from the front view, we have all of the branch tissue is now enclosed by growth from the trunk. So what this creates is an interlapping layers of cells, and that's what creates the strength of the attachment for a branch to the underlying trunk. So this image is showing an interesting phenomena that we see in plants related to their vascular systems. You would think that those xylem cells that are stacked on top of each other like straws and that are moving the water that's coming in from the roots would go straight up a tree. But we find that oftentimes that's not true. And what you're seeing in this picture is a spruce tree that's been damaged by a herbicide application that was made to weeds nearby. The tree has pulled up the herbicide, and it's causing some leaf yellowing. But what we find, interestingly, is that the damage symptoms spiral up the tree. They start from the bottom, and then they will spiral up the tree, not move straight up one side of the tree, as you would envision might happen with xylem if the cells were just stacked straight on top of each other. So it's just kind of interesting to make a note of that. One final thought on stems and that's related to the outer dermal tissue. So the term epidermis refers to an outer protective covering that can be found on any plant. It may include protective structures or hairs, as you see in this lamb's ear leaf, which is very soft and hairy. Or it may be, here's the lamb's ear right here, or it may be waxy, as in a jade plant, protecting the leaf from water loss when the plant grows in a drier climate. A periderm is a term that we find associated with a protective outer layer that we find on many roots and stems, which is made up of a corky type of a bark. It's formed by secondary growth within the stem. So in the picture of the crabapple above, we see the periderm, the outer bark, and again pointing to the fact that that vascular tissue is very shallowly located right underneath the bark. This tree's been hit by a mower in the spring when this tree was just starting to grow, and there was a lot of sap movement through the trunk. There's a flap of bark here that has been pushed away from the stem, exposing the underlying woody tissue and destroying the cambium, the vascular cambium within that section of the bark. All right, so finally, when we talk about plant morphology, the shapes of leaves and stems and roots and the flowers and the fruits that we may find on a plant, quite often we're looking at those things because we're trying to identify a plant. And let's do a little practice now. I've got some pictures that I want to talk about a little bit as far as how you might start going about determining what these plants are. So let's look at the first one. Okay, so let's say someone brings you something that looks like this. Now, how are you going to determine whether this is a compound leaf or this is a simple leaf? So is this one leaf right here, or is this whole thing one leaf? Okay, and the same goes for the picture over here. Is this one leaf, or is this whole thing one leaf? Well, to make that determination, you need to look at where, let's go back to this one, where this leaflet attaches to the underlying stem. So where its petiole attaches to the stem, is there a bud located right there? If there is no bud, then that tells you this is a leaflet, not a whole leaf. So going to this picture, we would look for the same thing. We would look to see, is there a bud in the junction of this little leaf structure? If not, we would go to the end of that main petiole and look right here. And in this case, yes, there is a bud here. So that tells us now that we have a compound leaf, not a simple leaf. So let's look at this first example. You may have a plant you're trying to identify that looks like this: yellow flowers, leaves, fruits. So a first thing that might come to your mind is, okay, well, we have a compound leaf. The bud for this leaf is located right here, right in the axil of the leaf. So we have a compound leaf that has many small little leaflets. That's one clue. Then if we're lucky enough to get a fruit, which we quite often are not, you might take a look at this and say, okay, well, this looks kind of like a bean pod. Okay, so we've got a compound leaf and we've got a fruit that looks like a bean pod. Well, the fact that we have a bean pod fruit would narrow this down enough that you could start looking at plants in the bean family. Okay, another clue might be that this flower kind of looks like a bean flower. Okay, so you could pick up a wildflower book and you could start looking at wildflowers that have a yellow flower, or you could look at plants that are located in the bean family and that might help you narrow down what this is. Let's look at another one. So here in this instance we have a weed, and we've got, I think the thing that would stand out for me most of all would be these seed heads. These seed heads look a lot like a dandelion seed head, don't they? And even the flower looks similar to a dandelion. Well, you might think, okay, well, what family is dandelion located in? Well, dandelion is in the aster family. So in this case, you might pick up a weed book and start looking at weeds in the aster family. And this happens to be Western salsify. Okay. Let's look at one more. So in this instance, we have a shrub, and the shrub has sub-opposite leaves. So they're not exactly opposite, they're not completely right across from each other, they're sub-opposite. They're kind of round, shiny. We've got these dark black fruits that develop in clusters. And there's one more picture. We have a stem that has a modified stem section that develops almost into a thorn. Okay, so those would all be identification characteristics that you could use to help you start to narrow down what this plant is. And this actually happens to be common buckthorn. Okay. We get questions about this one and whether the fruits are edible in the summer, and they are not. All right, so I have an activity for you to work on, and there's a couple of different other web apps that you can check out. So the Arbor Day Foundation has created a really simple tree identification key that you can use, and you can find that at the link that you see here. And it will take you through a plant identification key based on, what is the shape of the leaf, does it have flowers, and so on. For the major common trees, this would be a fun key to work through to identify a plant. Now, Virginia Tech also has an app that you can download on your phone that can help with tree identification. So it's called vTree, and if you'd like, you can check that out. Now, the one that I would really like you to try to experiment with a little bit is this app called PlantNet. It's a free app. You can download it from your Google Play or your Mac App Store. And you take a picture of the plant that you're trying to identify, whether it's the leaf, the flower, the bark, the buds, and it can help you determine and help you narrow down or make that identification a little bit faster. Okay. If you'd like to download the PlantNet app and give it a try in identifying some of your plants, I have a short video that will show you how to download the app and get started using it. You can find that video at go.unl.edu/plantid. I hope this little bit of a deeper dive into plant physiology and structures has been helpful to you. But if you have any additional questions, feel free to contact me at sarah.browning@unl.edu. Take care.